Good afternoon, everybody. How are you? I hope you're enjoying your beautiful day outside today. I hope you enjoyed day one's PowerPoint. Today is day two of DNA technology. You can continue along with your notes and we will begin. First off, we have three warm-up questions. What type of a virus can perform transcription backwards? What type of molecule ends in ACE, A-S-E? And what is the difference between blunt and sticky ends? So some very important questions from last time. Let's go ahead and see what you got. So here are the answers. Let's go over why these are the answers today. So the first question of what type of virus can perform transcription backwards? Those were retroviruses. Retroviruses were types of viruses that contained RNA rather than DNA. So R for retroviruses, R for RNA. And RNA, these retroviruses were tricky. Things like HIV, where it's so hard to cure because they don't operate normally. They can do what's called reverse transcription. They contain an enzyme, and the enzyme's name is reverse transcriptase. So this allows the virus to take its RNA, infect a bacteria or a cell, integrate its RNA into the cell's actual genome, into the cell's chromosome. So it could be a bacteria DNA, it could be your cell's DNA, but bacteria use reverse transcriptase as an enzyme that causes their DNA to become part of that cell's DNA. So whenever that cell performs transcription, it is going to make whatever the virus DNA says to make, which is usually make more viruses so people get sick. All right, next question is, what type of molecule ends in ACE? Do you know already? Enzymes. Helicase, DNA polymerase, restriction endonuclease, those scissors from yesterday that were cutting DNA. Any type of enzyme will always end in the suffix ACE or ASE. Clear those out. And the last question down below. What is the difference between blunt and sticky ends? So first off, those words describe the shape of how DNA was cut. So we've got a double helix, a nice double helix. If you have a blunt end, it's going to cut straight through that double helix. It makes a flat-ended cut. So both sides, this side and this side, have equal cuts. If I back up a little bit, a sticky-ended cut, this one's going to be slightly different. It's going to cause a single strand of DNA to be on each side of a sticky end. So there's going to be two sticky ends. So if we go up to our DNA and we make a cut like this, and then another cut over, we end up with a strand of DNA that also has a little single-stranded end. And then another piece of DNA over here that ends up having another single strand. These two ends were originally in the exact same spot. They want to stick together again. They want to be happy. They want to stick together. They're going to stick together according to Chargaff's rule. And if you remember, A will always match with T, C will always match with G. So our sticky ends match together with Chargaff's rule because they have that single-stranded part on one side. All right, let's go ahead and continue down. The agenda for day two of DNA technology is that we're going to talk about stage two. And in class, we would have had a plasmid matching activity, like a paper matching activity after. So the title for day two and stage two is also insertion into a vector. So we know we're going to insert something, and we see in the picture vector, but what is a vector? Let's define that first. All right, but it looks like before we define vector fully, we do want to reiterate that sticky ends are extremely important. So DNA from different sources, meaning DNA from 2 different animals, or a plant and an animal, or 2 different plants, can only be joined together. So the whole goal of DNA technology is mixing the DNA from different organisms. It can only be joined together if they have the exact same sticky ends. That means they would have to have the same recognition site. So if one animal has A on that little single-stranded part of their sticky-ended DNA, and another animal has T, you know what? Those are going to match together according to Chargaff's rule. You could combine those 2 organisms' DNA if they have the same sticky ends. And in order to get those sticky ends, you have to actually cut using the same restriction endonuclease, the same enzyme for both organisms. There's many different types of restriction enzymes, blunt-ended ones, sticky-ended ones. You'd have to use the same sticky end enzyme. And speaking of enzymes, look at that, endonuclease ends in ASE. That's an enzyme. So we have to have the same scissors, make the same cut on the same organisms so that we can mix DNA if we want. So these sticky ends, in order to actually glue them fully together, so let me take those lines off. In order to actually glue those sticky ends together, you have to use another enzyme. This enzyme is DNA ligase. It's going to act as a glue. Think ligase or linkage. It's going to be the glue that's going to hold those 2 sticky ends together. You can also visualize it in the picture down below, where we see a blue strand and a yellow strand being joined together, ligase. You can see that down here. And let's just double check. G, C, A, T, T, A, C, G. Yep, those would absolutely join together because they have the same sticky ends, meaning they were probably cut with the same enzyme. And when we combine organisms' DNA from 2 different sources, plant, animal, 2 animals, 2 plants, different things, you're combining it, the new DNA that contains, or well, the old DNA plus the new DNA, joined together and combined, we're going to call that recombinant DNA. Because we've recombined 2 completely different DNA sources together. All right, there we go. So that's recombinant DNA. Kind of an image here. This is supposed to be a GIF in the PowerPoint where you see it says target plasmid. And in this, of course it's not moving right now, but if it was, you would see that this restriction enzyme, it says restriction fragment, it would actually cut here, or no, I made a mistake. Sorry. Holy cow, I didn't know it could bring back the old stuff. All right. From the beginning. Restriction enzyme, there we go, would cut part of this out. Snip, snip, snip. It would cut it out. Then this new fragment from another organism, notice it's red, not black, it would be stuck in the exact place that was cut, and DNA ligase would do the gluing. There we go. So that's how that GIF was supposed to go. So again, a plasmid, which we're going to define in a moment, you can use an enzyme to cut it. If you cut it, you take a chunk out of it. You can use that exact same chunk, cut with the same enzyme from another organism, stick it in there, and DNA ligase is going to be the glue to join it together. There we go. So now let's define both of those. And it looks like we can't quite see plasmid. There we go. So we've got our next set of notes, insertion into a vector. So this is like the beef and potatoes. The main portion of the lecture is this part here. Let's finally define vector. Vector is literally anything used for transport. Examples of vectors: submarine, boat, ship, cruise ship, train, truck, a skid steer, cement truck, ship, blimp, helicopter, plane, all sorts of things. Anything used for transport is a vector. Next, the plasmid. The plasmid is this part, the little circle inside of a bacteria. It's not the bacteria's chromosome. It's just an extra little chunk of DNA that bacteria use. This plasmid is going to be the thing that can transport genes in DNA. So there we go, transport, transport. The plasmid can actually move in and out of this bacteria, and you can transport those genes that we've been cutting up from day one. And plasmids are pretty useful things because they contain antibiotic resistant genes. So that little portion in the bacteria can cause it to be able to not die in the presence of certain antibiotic drugs. Click back. All right, so this little plasmid, I mentioned that it had these antibiotic resistant genes. Let me switch to a different color. So some of these antibiotic resistant genes could possibly be broken or cut into in the process of those restriction enzymes cutting out sticky ends from genes. So let's look at the picture below. One plasmid, we can see that it has some different, it says RESA, RESB, RESC. It means just they resist, RES, they resist different antibiotics. So we've all been sick before, we've all taken certain type of antibiotics. Some bacteria have genes in that little plasmid that help them to not die in the presence of those drugs. So when you go to the doctor, it's good for them to know what bacteria are making you sick so they know which drugs to give you, which antibiotics that will actually kill the bacteria that they know they're not resistant to, which is very, very helpful. So let's continue reading. It says, to make room, oh, I crossed that off, sorry. So we want to make room for inserting the new isolated gene. I have a lot of annotations on there. So when we cut out a portion of that plasmid, we would take that portion out. Scientists want to make sure they are not cutting an antibiotic resistant gene. Because if they can grow up that bacteria and allow it to make a little bacterial colony, if it's still resistant to a certain antibiotic, the scientist will know, I didn't cut through that gene, which is very helpful. So for example down here, if you are using that plasmid, you took it out of a bacteria and you wanted to stick a gene into it that actually makes penicillin, that makes an antibiotic, because that's how scientists do create the antibiotics that we take now when we go to the doctor. There are bacteria that have this gene inserted into them that make antibiotics. And it's good if those bacteria don't die in the process. It's good if they maintain a resistant gene so they won't die. So you would want to put that plasmid into a cell that won't be killed by what it's making, the penicillin for this example. You would want to put it into a antibiotic resistant cell that is resistant to penicillin. And as I mentioned, scientists can take that, stick it into a plasmid where there are no resistant genes. They can make that bacteria create whatever was in the plasmid now, which is absolutely wonderful. So then the penicillin can be harvested and people can use it for medicine. All right, here's a picture of how that process would work. I'm a real visual person and I love pictures. So let's follow along. How do we insert a gene into a plasmid? This picture and this graphic is going to combine day 1 and day 2 today. So cut the gene you want out of the DNA to isolate it. So that's step 1. It says donor DNA here. Do you remember the example from day 1 with the person with red hair and the person with brown hair? Let's say we took some DNA from that person with red hair. use a restriction enzyme, the scissors, to actually cut just that gene out. And notice the sticky ends. They're uneven. They're like puzzle pieces. Next part. Get a plasmid. Take it out of a bacteria. Use the same restriction enzyme. So, using the same enzyme, cut that area out of the plasmid. Notice it's missing a chunk. And notice it is the same shape as this because we used the same enzyme. Step 3. Use ligase. Use another enzyme to glue that chunk in. Ta-da! We glued the chunk in. We've now made a recombinant or hybrid plasmid. Has DNA from 2 sources, from the original and from the bacterial plasmid. So in step 5, it says the bacteria can now make whatever the new gene coded for. So when that little plasmid goes in a bacteria are small and simple. They just read the DNA. And if there's new DNA in there, they read that as well. They're going to create whatever the DNA is. In this case of our example, it was, hey, from day 1, let's take a red-haired person's gene for red hair, put it into someone with brown hair. And maybe if you stick those cells in, that person could start, in theory, growing red hair. Not quite as simple as that, but for this example, it still works. All right, we are almost finished. That was day 2, pretty quick, quicker than day 1. However, we don't want to overload you with too much information at once. So let's end in a think, pair, share. First off, what is the importance of sticky ends? Hint, hint, that was our first slide. Second, what's a vector? New vocab word here. Give an example. We gave lots of examples during that lecture. And last, how can a DNA fragment be inserted into a vector? That was that graphic that we just went over. Give you guys a second to think. Oh, that's the activity. I didn't have answers to think, pair, share on my PowerPoint. Let's write them out. Here we go. So what was the importance of sticky ends? If you recall, the importance, there were several things that were important about sticky ends. First, you have to have a double-stranded piece of DNA that has a sticky end. Another piece of DNA with a matching sticky end. These would have to have the same letters according to Chargaff's rule so that they could be glued back together. The only way they can have the same sticky ends and the same letters to match by Chargaff's rule is if they were cut with the same restriction enzyme. So there's our importance of sticky ends. Next, what's a vector? A vector was anything used for transport. So what are things used for transport? We use them every day. Cars, buses, planes, trains. What was the example for transport we use today with gene technology? Started with a P. Plasmids. That circular piece of DNA inside a bacteria, we can use it to transport genes from one organism to another. Cut them out with the sticky ends, insert them into a plasmid. We can now turn it into a vector that can transport those genes to different organisms. And our final question, how can a DNA fragment be inserted into a vector? Well, first you've got to cut it with your restriction enzymes. Then you take that cut piece of DNA, find a plasmid, cut the same part out of a plasmid. Oops, I erased it. Time to start over. Take your DNA, use your restriction enzyme scissors, cut a chunk out. find a plasmid. Use those scissors to cut the same area out. Then use DNA ligase to glue it in. Now you have, that was supposed to be a star. That turned out crappy. Ta-da! Now you have a recombinant plasmid. It's got DNA from two sources, the original and the new. And again, the vector is the thing that is the plasmid, used for transporting that gene into another organism. All right, that was our quick day 2 lecture of how to insert DNA into a plasmid. I'll see you guys tomorrow or the next day for day 3 of our lecture. Have a great day. Enjoy the sun.